Hi, hello. This is Dr. Jagadish Narendra Prasad, Senior Lecturer in Botany, Sri Chaitanya Gosala, Vijayawada. Today, let us discuss about polysaccharides. Before discussing what are polysaccharides, first let us see about few details regarding carbohydrates. See, carbohydrates are basically three kinds: monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides, oligosaccharides, and polysaccharides. Let us take first monosaccharides. Monosaccharides are again two kinds: aldoses, ketoses, based on functional group present in them. If a monosaccharide with aldehyde group, such sugars are called aldoses. All monosaccharides are sweet in taste, they are water soluble, and those with the ketone group are called as ketoses. And based on number of carbons present, these are again different kinds like trioses, tetroses, pentoses, hexoses, heptuloses. Like that, we have different kinds of monosaccharides on the basis of number of carbons present in them. Trioses, the name itself is indicating that they have 3 carbons in their structure. So these are 3 carbon sugars like G3P, glyceraldehyde 3-phosphate, DHAP, dihydroxyacetone phosphate. These are all some important trioses. Tetroses, these are 4 carbon sugars like erythrose, erythrulose, 4 carbon sugars. Pentoses, ribose, ribulose, deoxyribose. These are all some important pentoses with 5 carbon sugars. Hexoses, 6 carbon sugars like glucose, fructose, etc. Heptuloses, 7 carbon sugars like pseudoheptulose, These are heptuloses. These are all monosaccharides. Let us see about oligosaccharides. Oligosaccharides are those with 2 to 9 monosaccharides linked by glycosidic bonds. Around 2 to 9 monosaccharides are linked by glycosidic bonds in a oligosaccharide. These oligosaccharides are also again different kinds like disaccharide, trisaccharide, tetrasaccharide, pentasaccharide. These are all different kinds of oligosaccharides. Disaccharide is one with 2 monosaccharides linked by glycosidic bonds. There is one glycosidic bond between 2 monosaccharides. We have some examples for disaccharides like sucrose, maltose, lactose. These are all some important examples for disaccharide. Similarly, trisaccharide, raffinose, tetrasaccharide, stachyose, pentasaccharide, verbascose. These are all some important oligosaccharides. Now let us go into polysaccharides. When it comes to the polysaccharides, along with the nucleic acids, proteins, polysaccharides are also got separated into acid insoluble fraction. Because polysaccharides are again macromolecules with more than 9, means around 10 to many monosaccharides are linked by glycosidic bonds. We can compare this polysaccharide with a thread like cotton thread. So they are long chains of monosaccharides. All polysaccharides are long chains of monosaccharides. Polysaccharides are divided into two kinds on the basis of number of types of monosaccharides present in them. If a polysaccharide has only one kind of monosaccharide, it is called homopolysaccharide. Whereas if a polysaccharide has more than one type of monosaccharides, heteropolysaccharide. Homopolysaccharide contain only one type of monosaccharides. Many monosaccharides, only one single type of many monosaccharides are linked by glycosidic bonds in homopolysaccharides. In heteropolysaccharides, more than one type of monosaccharides are linked by glycosidic bonds, so that they are called heteropolysaccharides. Let us take some examples for homopolysaccharide and heteropolysaccharide. First, let us see about homopolysaccharides. When it comes to the homopolysaccharides, cellulose is one important homopolysaccharide. Cellulose is made with beta-D-glucose. Many beta-D-glucoses are linked by glycosidic bonds and forms a thread or a chain of glucoses that itself constitutes cellulose. So, cellulose fiber or thread is made with beta-glucoses. Therefore, in cellulose, you could see beta-1,4 linkages, 1,4 glycosidic linkages. The second example, starch. Starch is a variant of cellulose. It is a variant of cellulose. And starch is made with alpha-D-glucoses. Alpha-D-glucoses. Only alpha-D-glucose is present in the starch. And starch is said to be storehouse of energy in case of plants. In plants, the energy is stored in the form of starch. Another variant for cellulose is glycogen. Glycogen is also made with alpha-D-glucoses. Glycogen is also a homopolysaccharide and it is storehouse of energy in animal cells. Storehouse of energy in animal cells is glycogen. Storehouse of energy in plant cells is starch. Another example for homopolysaccharide is a chitin. Chitin is another homopolysaccharide. So, chitin is composed of N-acetyl glucosamine. N-acetyl glucosamine. So, these are some important examples for the homopolysaccharides. We have another example also like inulin. Inulin is composed of beta-D-fructoses, beta-fructose. It is a homopolymer of the fructose. Inulin is a homopolymer of fructose. Remaining all are the glycogen, starch and cellulose are homopolymers of glucose. Chitin is a homopolymer of N-acetyl glucosamine. We'll discuss about this N-acetyl glucosamine at the end of our topic. Then let us see about the heteropolysaccharides. We have two best examples for the heteropolysaccharides. One is pectin and the second one is hemicellulose. Remember, cellulose is a homopolymer, hemicellulose is a heteropolymer. Pectin is also a heteropolymer. Apart from this, on the basis of functions, these polysaccharides are grouped into three categories. Number one, storage polysaccharide. Second group, structural polysaccharide. And the third group is a mucopolysaccharide. Mucopolysaccharide. These are the three important groups on the basis of their functions. Storage polysaccharides include starch, inulin, glycogen. These are the best example for storage polysaccharides. Let us take first starch. As we discussed earlier, starch is made with alpha D glucose. Starch forms secondary helical structure. Secondary helical structure. Starch has amylose and amylopectin. This is the second helical structure with amylose and amylopectin. The secondary helical structure can hold I2. Therefore, starch with iodine gives blue in color. It gives blue color. Whereas cellulose doesn't form the secondary helical structure. Therefore, you don't see any kind of blue color when you treat cellulose with iodine. Whereas starch gives blue color when you treat with iodine. And inulin, as we discussed, it is a homopolysaccharide of fructose and it is a storehouse of energy in case of some Asteraceae members like Dahlia, Dahlia, Asteraceae members like Dahlia, Helianthus. In such plants, inulin gets stored either in the roots or in the stem. Say for example in Dahlia, roots store inulin. Helianthus stem store, underground stem store inulin. When it comes to the glycogen, this is the structure of glycogen. Glycogen is composed of alpha D glucoses. Now this linkage is called alpha 1,4 linkage. 1,4 linkage, whereas this linkage is called alpha 1,6 linkage. That means glycogen, like starch, glycogen is also branched. Starch is branched, glycogen is also branched. In the branching region, you could see 1,6 linkage, 1,6 type of glycosidic bond. Whereas in unbranching region, there is 1,4 linkage. When glycogen is treated with iodine, it gives red color, not the blue. Glycogen with iodine gives red color and it is the storehouse of energy in case of animal tissues. In animal tissues, it is the storehouse of energy. And in any polysaccharide, there are two ends. Left end is called non-reducing end and right end is called reducing end. Right end is reducing end, left end is non-reducing end. Means in the right end, reduction reaction takes place. Therefore, it is called reducing end. On the left end, no such reduction reaction occurs. Hence, it is called non-reducing end. So, these are some important storage polysaccharides. When it comes to structural polysaccharides includes cellulose, hemicellulose, pectin, chitin. These are structural polysaccharides. Let us take first cellulose. Cellulose is one of the most abundant organic substance on this biosphere and it is cell wall component of all plants. It is also seen in some fungi. It is a cell wall component. And as we know that cellulose is made with alpha, beta, D glucoses. It is unbranched. It don't form any kind of secondary helical structures like this. So it doesn't react with iodine. So that cellulose with iodine doesn't give any kind of blue color. Cellulose is present in the plant cell walls. Paper pulp is made with cellulose. Cotton threads is made with cellulose. That is the importance of cellulose. Hemicellulose and pectin both are heteropolysaccharides. They are also cell wall components of plant cells. Hemicellulose is present in the cell wall. Pectin is also present in the cell wall. Pectin is most abundant particularly in middle lamellum of cell wall. That is the importance of pectin. And some polysaccharides have building blocks like amino sugars and chemically modified sugars. Remember, some polysaccharides have amino sugars and chemically modified sugars. Chemically modified sugars like glucosamine, N-acetyl galactosamine. These are chemically modified sugars. Glucosamine is an amino sugar. Amino group is attached to the sugar. When amino group, NH2 group is attached to the sugar, it is called amino sugar. Or sometimes they may be chemically modified sugars. So such amino sugars and chemically modified sugars becomes building blocks for the organisms. One such example is chitin. So chitin is composed of N-acetyl galactosamine. It is a chemically modified amino sugar. And chitin is a homopolysaccharide. It is a complex polysaccharide. Complex polysaccharide and homopolysaccharide, and the building blocks of this chitin is N-acetyl galactosamine. This is another most important thing we have to remember. The last one is mucopolysaccharides. Mucopolysaccharides are heteropolysaccharides with high molecular weight. They have building blocks like galactose, sometimes arabinose is also present, mucinic acid is also present, polysulfate esters are also present. So these mucopolysaccharides, they are with high molecular weight and heteropolysaccharides. The best examples, agar is present in red algae. It is a polysulfate ester, red algae containing agar-agar. Agar-agar is obtained from Gelidium and Gracilaria, and it is used for making ice creams, jams and jellies, and it is also used as a solidifying agent for making microbial nutrient medium. Apart from that, you can also see in carrageen. Carrageen is also another important cell wall component present in red algae. It is a phycocolloid or hydrocolloid present in red algae. These are the best examples for mucopolysaccharides. So mucopolysaccharides are heteropolysaccharides with high molecular weight. So this is what you can see with regard to polysaccharides on the basis of their functions. So on the basis of functions, we have three categories. Storage polysaccharides like starch, inulin, glycogen. Structural polysaccharides like cellulose, hemicellulose, pectin, chitin. And mucopolysaccharides like agar-agar, carrageen, heparin. Heparin is also a mucopolysaccharide. These are all some important examples with regard to the polysaccharides, and this is about few important things which usually asked in the NEET examination about the polysaccharides. We'll meet again in another video. Thank you very much.